All right, good morning. My name is Molly. I'm the research leader, Cereal Crops Research Unit, Madison, Wisconsin. Today, I'm really excited to talk to you about abiotic stress resiliency in barley. This is a project that I started when I joined the USDA in 2015, 2016. Let's see, oh, this is going. Okay.
Is it not advancing, Molly?
It's not advancing.
Done.
That's the one I'm trying. I can see it on the next slide, but it's not advancing.
Perhaps try that.
Okay.
Is that good? Okay.
See. So I talk a little bit about climate change because this is about climate resiliency, particularly heat and drought. Try to summarize 4 different approaches that we have been taking to address this issue, starting with phenotyping and mini-core collection, a old-fashioned traditional QTL mapping-based approach, and a transcriptome-based approach to understand some of the tolerance mechanisms that could be operating, and some updates on some transgenic work that we initiated recently. Hopefully, I would like to spend a little time talking about some perspectives I gained working on this project over the years in the context of climate resiliency. So I've been touting this one for some time now, that when you have combined stresses or concurrent stresses, it's much more damaging than single stresses. People are just focused on looking at only drought or only heat or, wheat haphid or blast or whatever, one factor at a time. This is a very reductionist approach, and this is probably not going to work out well, especially when you're talking about improving climate resiliency. We'll talk more about this in the end. This is a meta-analysis that I did as a part of the book that we wrote way back in 2015 on combined stresses in plants. So taking into consideration all the billion-dollar naturally occurring disasters for the past 3 decades, starting from the year 1981 to 2011. And it is found that whenever you have a combination of heat and drought stress, actually the billion-dollar yield losses that you can see there is much more exaggerated when you than comparing only the drought stress or flooding stress alone. Okay. And we put out a review article recently taking into consideration both biotic and abiotic stresses in 2021. The story is the same. And I also want to show you another study that we've been doing. I want to thank Scott Eisel for sharing this data. This is just for barley, data collected based on crop indemnity losses paid out to the US barley growers between the years 2013 and 2023. And you can see about $32 million was given out for heat-related losses, and about $140 million. This is just in the United States alone. So this is an issue. This is important. This is happening. And then I did a little correlation study about how many cases were there for heat and drought, and it looks like there is a 94% correlation, suggesting that what we are seeing here is the combination of heat and drought. To give a best example, I think 2021 was a bad year for barley, right? I think $57 million was made out in 2021. So this is all about messaging. I always want to put this slide out. I know it is redundant at this point in time, like Brian mentioned yesterday, but especially for the students, we can write review articles about heat and drought, climate change. People don't listen to it. But you put out a figure like this, drink up your barley, then you're getting calls from NPR, USA Today, everybody wants to interview you, okay? So this is a very theoretical paper, just based on climate projections. They come up with this idea how much will the barley be costing, and in turn, the impact on the beer prices, okay? The most important thing for me here is to point out, it's in small text there, it is during years of drought and heat waves, okay? It's not drought or heat. It's this combination, okay? We need to take that into consideration when we are talking about climate change. So this is the only technical slide I will show you, but I think this is very, very important for people to understand. Doing this kind of heat and drought stress screening entails a lot of planning. If you're thinking about it, please talk to people who have done it. Pay attention to every little detail, especially if you're going to set it up in a greenhouse setting. You want to pay attention to every little detail that you can, including the pot size that you want to use, the soil mixture that you will be trying to use, and keep it consistent across your screening, your applications, if you want to get something meaningful out of all of that study. This is about 3 years of work, screening thousands and thousands of parts. And then when you talk about temperature stress, we are not just throwing them into a chamber at 30 degrees and say this is heat stress. That's a heat shock. So the way we program this growth chambers is that they increase by 1 degree centigrade every 15 minutes. So it reaches the max at 30 degrees or 32 degrees by midday, and then you slowly ramp it down by 1 degree every 15 minutes. So coinciding with the light and dark cycles and maintaining that. So that's very important. And the soil mix determines the field capacity or how the water holding capacity, and you need to understand your soil mix very well to impose this drought. We monitor the pot conditions every day and record the soil moisture content. Barley is kind of drought tolerant, if you will. It can go down to about 10% of the pot holding water holding capacity, and it will survive. Okay, we don't want to stress this too much because we do want the plants to set seeds and give us some data. So you don't want to not water them at all. That's some studies where I have seen people just, oh, we withheld the irrigation for 15 days. No, that doesn't work. No, it has to be realistic. Okay, so this is what we do in our screening experiments. It's about 5 days. Day 0 is our ramp up day. We do the stress treatment for 1, 2, 3 days. 4th day is a ramp down. 5th day it comes back to normal temperature and watering regimes. So the next important question to ask is, what is the best stage to screen these barley plants, right? It will be very easy to set it up in a especially in a greenhouse setting. If you want to do thousands of plants, maybe do the seedlings, it's the best way to do it. You can screen thousands of plants very quickly and get done with it. So we took up a small study in collaboration with Phil Bregitzer, about 5 varieties, and did this under 2 different stages. You can stress them at the vegetative stage. This is about 3 to 4 weeks old. And then you can go through this whole exercise, and we also did the malt quality analysis. So when you stress them in this vegetative stage, yeah, the plants look a little bit sick, but they recover very quickly. And there was no difference in the quality of the malt profiles between these plants, which were stressed and non-stressed when the stress was imposed during the vegetative stage. On the other hand, you hit the same stress during heading stages. Okay, boy, it's a big difference. There was a big fluxes in all the photosynthesis parameters that we evaluated. And then the malt quality profile looked terrible. In fact, this paper in Physiology Plantarum was selected as one of the best papers. And this cartoon comes from their, what is that, cover article they cited us for this work. So we did this work with all the amber malting barley varieties, and we found that none of the current varieties which are cultivated in the US have any level of tolerance to combined heat and drought stress. But if you hit them singly with heat or drought, you will find some different levels of tolerance to that. But if you put heat and drought together, about 95% of the yield is lost, and the amount of seeds we get, and even to do this small mini scale malting that we do using T-balls, the malt quality profiles look terrible. So what is the best resource for finding heat and drought tolerant germplasm? So this was when I was just starting at the USDA, I found this really nice piece of work by Gary's group. They published this ICOR collection, genotyping more than 2,400 accessions using the 9K chip. very nice piece of work, and it gave me some insight into this wonderful germplasm that is out there. And thought maybe this is a good place to get started for screening. We have some data here, and but 2,400 accessions is a lot of accessions for us to screen, given our resources and the size of our greenhouses. So we had to come up with some smart way of picking the best subset, if you will. And in fact, in that paper, they talk about using the polymorphic information content, where they are taking out one line at a time and seeing how it affects to the average polymorphic information content. That way, when you pick the top 10% of the lines, you probably capture the maximum genetic diversity within this pool. And that was the idea behind this. And when we did this, we ended up with about 186 accessions. And we went on to do a 50K SMP chip, and in collaboration with Brian Stephenson Ahmad, we did the structure analysis. You can see the principal component analysis plot here showing that we got a very similar ancestry spread like what we saw in the previous slide, suggesting that we really have a good subset of this I-core collection that we can use now. And within this, we call this the mini core, and there are 166 spring barley lines in the 16 winter types and 3 facultative lines. Since we are talking about heat and drought stress, we paid attention only to those 166 spring barley lines. So we did this barley screening of these 165 lines at the University of Wisconsin Biotron facility using the setup that I described to you a few slides earlier. This was a very long endeavor with the pandemic and government shutdown for 45 days, followed by people leaving. We somehow got it done, 3-4 rounds of screening. We got this whole population screened for singly applied heat stress and drought stress, and a combination of heat and drought stress. Should tell you, it's very challenging to organize these experiments because the range of heading dates spans anywhere between 71 to 129 days, with the median being about 110. So we want to do all our stress experiments over that 1 week period when they start heading. So we had to do a lot of analysis beforehand to stagger the planting, because we had a good data on the heading dates. So we would stagger the planting such that the plants would come in batches, so we can accommodate them in the growth chambers to do our heat and drought stress experiments. Okay, the long and short of this story is that, well, more than 100 accessions, you can say that they just are extremely sensitive to a combination of heat and drought stress. But we did find one line which seemed to be holding up very well. We call this line number 136. I'll tell a little more about it. And we are pretty happy that this screening at least gave us something. Line 136, we also sent it out to Aberdeen, and it has been planted both in the irrigated and the rain-fed conditions. And we are actually doing the malt quality analysis of this right now, even as we speak. And my postdoc has already done some transcriptome proteome analysis, and also looking into some microbiome aspects of this particular line. So line 136, it's a line accession coming from Czech Republic. It's a commercial cultivar. The name of that is Malvus. I don't know if anybody know about this particular one. Malvus is a malting variety, and it was stouted to have some kind of a BAMI1 thermostable allele. That's another project that we are working with Marcus Vinci, another scientist in our unit. So that's where this is. So this is the first project doing a phenotype screening, just looking at shoot and root biomass and seed yields, and trying to understand, identify this germplasm, trying to further characterize it. And we'll be happy to share this line with breeders if they are interested. It is showing promise, I think, in the field based on the data I got for 1 year. I'm still waiting on the data from Montana to come in. We lost in the rain-fed condition. We got about 84% of the yield compared to what it was in the irrigated condition. So it's pretty good. So now switch gears, talk about 2 other projects and related projects. One of them is looking at this heritage barley varieties like Otis and Golden Promise. So when we did all the screening for heat and drought stress, I realized that all of this work was done using malting barley varieties. So I was like, well, maybe there is something out there in the feed barley varieties, because I know there has been a lot of history in terms of feed barley variety development for dryland conditions. So there must exist some germplasm in the US collection somewhere, which is tolerant to heat and drought. And this is when I met Dr. Phil Bregetzer in 2016 in my first big conference here. And he said, like, I've been waiting to see somebody like you. Like, I have this awesome line. It is called Otis. It's a feed barley varieties. You can stick it in the oven and it will survive. I was like, wow, like this is that heat tolerant. Okay. And then Phil had this interesting story, I believe in 2008 or 2009, when there was a severe drought. He thought his entire field experiment was done. And as he was driving back home, in his peripheral vision, he caught sight of one plot which stood out. It was still green. And he went back and saw it was all this Otis plot. It was the only one which survived that year. So he fell in love with Otis that time, and he started making crosses, and he has been, and he was very generous to donate all of these things to us. So as you can see, and this is just a representation of how the seeds look like between GP and Otis. GP is Golden Promise, and HD is the heat and drought stress. You can see that this is just a random sample of 10 seeds to show you guys like how bad the seeds look in Golden Promise compared to in Otis, which looks pretty okay. So, like I said, Phil has developed this RIL population using this. He gave it to us. It's about 190 RILs in it. We genotyped it with PK. We have also done some drought phenotyping in the greenhouse conditions, and we also did 2 seasons of both under irrigated and rain-fed conditions, and did some QTL mapping. Again, this is published work. I'm not going to delve too much into it, but I do want to share a few things. One of the things is that we found a very nice QTL associated with shoot weight. It's actually the QTL, it's very close to a gene called the ascorbate peroxidase, or APX. And it is about 850 base pairs in the promoter region of this gene itself. So that was pretty exciting for us. And the other thing is, this is the marker effects you can see. So the plants which have the GP allele, which is shown in the left in the green color, okay, that's the one which has higher shoot weight. And on the right side is all the genotypes, all the RILs, which has the Otis genotype for this particular marker. So you can see the difference in the shoot weight. The median shoot weight is 72 versus 41. So why is this very exciting? Because shoot weight is one of the very important characteristics that impacts the seed yield. And it has been shown that during terminal stresses like heat and drought, especially, the shoot weight plays a vital role, because all the assimilates from the shoots have to be translocated into the sink tissue. So plants which have the ability to translocate all of their assimilates from the stem into the seeds, they help in overcoming this seed yield loss, and that's what we see in Otis. And while on the other hand, we have a higher shoot weight in the Golden Promise, suggesting that maybe it is not able to move all of those assimilates successfully into the seed. So there is a poor grain field and hence lower seed weights. And so this is very nice. It fits in with what has been observed in wheat and also many studies in barley before. Like I said, this is a published piece of work. I want to tell why this particular enzyme is exciting from a biochemistry point of view. I want to tell a little bit about this ascorbate. APX stands for ascorbate peroxidase. So it's an enzyme, and it is very important for detoxifying reactive oxygen species, which is usually produced in response to any kind of biotic or abiotic stresses. Okay, so ascorbate peroxidase takes reduces the hydrogen peroxide to water, and in the process it itself gets oxidized to become the MDHA, which is monodehydroascorbate, which is very unstable, and it will spontaneously oxidize itself into dehydroascorbate DHA. Okay, So there is an enzyme system for recycling this ascorbate back, which is called as DHAR, which is a dehydroascorbate reductase. And there is also a monodehydroascorbate reductase. And in this process, the DHA is converted back into ascorbate. For those of you who don't know what ascorbate is, it's just your common vitamin C. Okay, so in this process, you can see it uses glutathione. So the glutathione becomes oxidized to GSSG. And again, there is another enzyme called GR or glutathione reductase, which will recycle back the oxidized glutathione to reduce glutathione. So this is a typical ascorbate recycling pathway. I'm showing this because ascorbate biosynthesis itself is a very expensive process. So plants have developed systems, innate systems to recycle the ascorbate as they get oxidized. And APX plays a very important role in this process. So it's pretty cool that we have this. And even more exciting for us was the fact that I looked at the structure of this particular protein. You can say this is the ascorbate binding pocket, which you can see there in that orange color. And there are 3 conserved amino acid residues, here proline 63, aspartate 75, tyrosine 97, which has been shown to be a glutathione binding pocket. Okay, and so this is a kind of a cool enzyme which can take both ascorbate and glutathione. And it has very strong implications on what happens to that ascorbate and the glutathione pool. Okay, so if you follow the logic behind that pathway, and if you have a dual substrate binding enzyme like this APX enzyme, what it means is that your pool of reduced ascorbate will be higher, and also the pool of the oxidized glutathione will be higher. And this is very important because these redox molecules are important biochemical cues which promotes phase transitions. What does that mean? Phase transitions means those are signals which tell the plant, you need to flower now, you need to set seeds now, things like that. These are all things which we learned from looking into model plant systems like Arabidopsis. I don't know if this is still holds good or not in this system, but we are pretty excited that we have a dual substrate binding enzyme, and this is closely associated with drought stress. And it makes a lot of sense in the context of where we are looking at it. So this is a review article I came across in a pretty unknown journal. This is Shendao Lee's a group from Murdoch University. They put out this really nice paper on potential candidates for gene editing and molecular weighting for improving abiotic stress tolerance. And I just want to point your attention to chromosome 2 on the top. You can see that is HVAPX2 is the same gene that we are talking about here. And they say that it is associated with drought and cold and salt stress. And even more exciting for us is the fact that it is in close proximity to this PPDH1, okay? In the context of the phase transitions that I mentioned to you, the fact that there is a developmental gene associated with this flowering and timing in barley is pretty exciting. And it seems to all fit in very nicely in the story. So that's what I wanted to talk about here for that mapping project. We also undertook a very extensive RNA-seq analysis. I think I've talked about this in the previous book, but I will point out some new things that we think is relevant in the context of climate change here. So we did RNA-seq on using flag leaves and also the head tissue 1 day and 5 days during the stress. And this is in GP and ODIS. We did this just with heat alone, with drought alone, and in a combination of heat and drought. And what you're seeing on this chart is just the number of differentially expressed genes, how many genes are going up and how many genes are coming down. If you pay attention to the middle of the panel where you can see drought, 1 day, not a whole lot is happening in terms of gene expression changes, but at 5 days into the drought, you can see thousands of genes are up and down. Again, the purple bar is the golden promise, which are up, and then golden promise down. What is up and what is down are in red and yellow color. Okay, so I won't go into the details of this. You can see in initial response, transcriptomic changes in response to drought is very minimal, and then it all shoots up in 5 days. While in response to heat stress, some big changes happen early on, but not to the extent that you will see on the right side of the panel, which is the combined heat and drought stress. The point of this is that plants perceive heat and drought stress as a totally different stress or phenomenon. Okay, it is not just an additive effect of heat or drought alone. So the massive reprogramming that happens in response to heat and drought stress is the take home from this. And there is second take home message. You can see, I don't know how many of you can see the numbers out there, but the change in the gene expression in golden promise, the sensitive line is much more higher compared to in Otis. So we thought, okay, maybe Otis is probably inducing those genes to very high levels, and those are the ones which are important. But when we screened through this list, what we found was that there was nothing unique in Otis that was not in golden promise. So it was the same set of genes that were up or down regulated. And even more surprising was the fact that the amount of induction was much higher in the sensitive plant than in the tolerant Otis plant. Okay, this was like, wow, this is not what we had expected. But it led us to a very interesting hypothesis. If you think about the biotic stresses, we always talk about innate immunity. So we thought like maybe there is something innate tolerance built into Otis. So it really has a higher threshold. So you just have to not work as hard as GPA. You can just tweak it by 1.5-fold and you're fine because you're already at a higher threshold. We were in a very good situation to test this hypothesis because we have controls for every one of these tissue time points. So we had like 48 libraries from which we could take and look at this data step by step. And again, we did this using a pathway enrichment analysis, the KIG pathway enrichment analysis, just to get an idea of is this hypothesis true? And if so, what are the genes which are already up or higher in the non-stressed Otis plants? And we took very stringent criteria for this. At least it should have 20 counts, and I know the log 10 p-values, we set it to very high. We don't want any false positives in this process. And I'll just give you a little overview of what we found. So these three pathways, I just will briefly talk about this. Again, the theme is, redox homeostasis seems to be the key for handling all this. Glutathione metabolism comes up. Okay, what is so exciting about it? Again, take a look at the genes. I just highlighted ascorbate peroxidase. We talked about it there is this enzyme, dehydroascorbate reductase. We talked about this in the ascorbate recycling pathway. And then there is gamma glutamyl cyclotransferase. This is one of the key enzymes important for GSS biosynthesis. So innately, Otis seems to be having higher levels of expression of these key genes, which are important for maintaining that good redox balance, if you will. Okay, coming to starch and sucrose metabolism, exokinase. There is lots of papers out in Arabidopsis and on other model systems, even in rice, that if you overexpress exokinases, it really helps the plant to tolerate a lot of oxidative stresses. And trehalose phosphatase is a key enzyme which is important for the biosynthesis of trehalose, which is an osmolyte important for drought stress tolerance. So again, these are all. So the yellow one band there, it shows how much higher it is in Otis compared to GP. The negative sign is because I'm looking at the GP to Otis ratio here. So that means it is 2-fold or 3-fold higher in Otis compared to GP. Okay. And in the bottom, what we see is alpha-linolenic acid metabolism. Again, this allene oxide synthase and the peroxisomal acyl CoA oxidase. These are key enzymes involved in the biosynthesis of jasmonic acid, which is a pro-life molecule. Okay, this is an important hormone. And so that's very exciting to see. So there is this concept of innate tolerance. So it does make sense. So just don't disregard your feed barleys and take a look at it. Maybe my hope is that at some point in time we do this kind of transcriptome analysis so we can create a pan-transcriptome database. So where we have looked at different tolerant varieties, so we can identify novel mechanisms of tolerance and see if we can up it in our commercial varieties. So this is the last part. I'm going to talk a little bit about this project where we are trying to develop a transformation system in collaboration with the Wisconsin Crop Innovation Center. This is a collaborative project between ARS Aberdeen and CCRU and the Crop Innovation Center. The idea is that to date, only Golden Promise has been most successfully transformed in barley. We were trying to see, can we develop a system where we can transform commercial barley varieties. So we just took Gemcraft for this particular project, and Conlon also was in the fray, but we decided that we can focus just on Gemcraft for now to show the proof of principle. So at WCIC, these guys have perfected this technology for isolating mature embryo explants, and then using that meristematic tissue leaf base to induce multiple buds, and then ways of regenerating roots and shoots from them, and hopefully get a successful plant. So they have done this, and it took them more than a year or a year and a half to perfect this. They were successfully able to transform. What you're seeing there in the second panel is both the GUS and GFP was used as a proof of principle to show the transformation is working efficiently for them. And in the end, what you're seeing is the Gemcraft plants, which was transformed with GUS. So in the proof of principle, we know that it is working. On the right side, what you're seeing is the GUS staining of each of the leaves, because one of the things about this method of transformation is the chimerism. So they wanted to show that every leaf is staining. There is no chimeric effects. That means only a few of them, meristematic cells, get transformed. So it won't be a stable, it won't be transmitted to the next generation. So they were at least able to show in this one or two events that yes, they were all positive, and it was also verified by a PCR approach. So this is great. We have a transformation system which seems to be working well, and at least in principle. Now we need to select some target candidate genes to go after. Okay, so this is, what genes do you want to target for improving climate resiliency? This is not an easy question to answer. Okay, like to think about it, it's why don't you pick ascorbate peroxidase? I would say that's a dumb idea. Okay, because I know about this enzyme, and it also, you don't want to use these kind of enzymes because it can have repercussions in terms of how they look in terms of phenotype. I mean, in Arabidopsis, they have done it, and usually it results in a stunted plant. When I talk about Arabidopsis, people make joke about it, like, anyway, Arabidopsis is stunted, like, why are you worried about it? Okay, no, but if you, in the context of barley or any other crop, that's something we have to keep in mind, like, how does it affect the phenotype? So I would like to focus mostly on transcription factors. I think there are better targets to go after. I'll give you some more evidence for why that is the case. And I just picked this one. This is called the ATHB, a very well-known gene family. It's a homeo domain leucine zipper family of proteins. The reason I'm excited about this is there's tons of paper out there in model systems like rice and Arabidopsis showing that it is important for redox homeostasis. Okay, and you can see this ATHB7 and ATHB12 there on the right side of this matrix are glowing in red. Red means high levels of expression, and green means low level of expression, and black means no change in gene expression. Okay, so they have done this using rosettes and flowers and in seedlings, in PEG media and things like that. So this seems like a really good one. The question is there evidence of this in barley at all? Like, yes, barley has about 26 members in this particular gene family, and they have shown that many members of the gene family are induced in response to drought and also in response to cold stress and salt stress. So, looks like this would be an ideal candidate, ideal gene family to look at. But one more reason why I was interested in choosing something like this is I have a lot of biochemistry intent, so I want to show this little cartoon here and explain what is going on. So this protein can act as a homodimer or an heterodimer. Okay, well, I'm just showing the picture of a homodimer. That means there are two copies of this protein, and the leucine zipper, it is like a zipper. Okay, it hybridizes these two units together, and the homeodomain is the part which recognizes the DNA. And on the C-terminal end of this protein is what is called as the activation domain. A transcription factor has a DNA binding domain and an activation domain. And the activation domain is what is called a AHA motif, okay? The AHA motif is very important because it is full of aromatic amino acid, hydrophobic amino acids, and acidic amino acids, which forms very tight interactions with the RNA polymerase basal transcription machinery. So targeting transcription factors which directly associated with the transcriptional machinery are better targets, in my opinion, than choosing transcription factors which are not associated with the transcription factor. This is something we can talk offline, but this was another reason, rationale behind choosing this particular family. We can talk about this, like I said, offline. I want to get into the details of this choice. But is there any evidence? Okay, is there any evidence for this particular family of transcription factors? There are two wonderful papers that came out. One in wheat, where they express this HAHB4. It's a sunflower version of this HB4 transcription factor. And they did like 7 years of field study and 37 different locations. This was a study in Argentina, and it is now approved to be grown in Argentina. I think recently it was also approved to grow in the US. And then the next success story from the same transcription factor, this comes from soybean, which was again approved in Argentina. They show that this was very exciting for me because it's in warm and dry environments in Argentina that they show that this plant is doing very well. So these are two commercial successes of HB4 gene itself, and which got me excited. So we overexpressed the HAHB4 in GemCraft, and we used the maize ubiquitin promoter to do this. And I'll just share one or two pieces of data and then wind up here. So we are screening the T3 generation plants for these events. I just picked 3 independent lines. We got about 12 events, and some of them were chimeric, and then I just selected the homozygous 3 independent events. But what I'm showing there is the highest yield I found in these plants, and the bottom bar shows the range we got. So it seems to be out yielding. These are just standard normal greenhouse conditions. We got this 20, 30, 40 grams per plant increase in seed yield. Okay, they have not been stressed in any way. This is just normal growing plants, but they look beautiful. And when you overexpress some of these transcription factors, you get really weird phenotypes sometimes, but we were not. They just looked exactly identical to a non-transformed GemCraft. And very quickly, we also did a germination test. The ones on the left with the broken lines are just done with water. You can see it's a faster germination for the tree transgenic lines within 1 day. And actually, it may be much faster because they look a lot bigger in 1 day. We want to do a shorter time course. And on the right side, what we have is polyethylene glycol. It's a compound that people have been using for screening for drought tolerance at seedling stages. We have been doing this for the mini core and also for the RIL population. I'm not showing any of the data here today, but when we did the germination in presence of PEG, we found that the transgenic lines were doing pretty good compared to GemCraft, which is shown in blue and wow. So we're pretty excited about this. Looking forward to doing the drought and heat and the combined stress screening. So with that, I would like to conclude. These germplasm collections are really valuable going for identifying normal, abiotic stress tolerance. And then don't forget about feed barley varieties. You can learn something from them. And if you or anybody knows some interesting germplasm that is tolerant to abiotic stresses, please let us know. We registered this population. We also dumped all the data into T3 so people can use the GP-ORDIS RIL population for screening for any other traits if they are interested. And I think APX gene is a viable target for marker-assisted selection for improving drought tolerance. And we are looking forward to screening these transgenic lines that we have got for drought and combined stress. Hopefully, we'll give you an update in the coming meeting. But I want to end in the context of climate resiliency, and also maybe will act like a segue for the discussion session. Ashley, if you don't mind, just 1 second. As we think about climate resiliency, I think we all need to broaden the horizons. This is not just heat or drought. I hope, I was thinking that Scott mentioned this is 44th EMBA big meeting. So my vision, I see it's like 46th, 47th meeting when we meet here, we are not talking, there is no session on RWA or a BLS or a heat or a drought. It will be a more holistic approach. Only then we will be able to get something viable to the grower. Okay. Climate change is happening, it is real. Okay. And how we address this depends on how we come together as a group, a community, and it's not about a pest initiative or a scab initiative. It has to be a holistic initiative. Okay, it has to be people coming together, putting. So if you are screening for heat and drought, you are also screening for RWA, you're screening for BLS, and if there is a nice idea there, agronomic practice to beat PHS, yes, we would like to know. Need to incorporate all of those kind of things if we want to provide some viable solutions for the future. I'll leave with that thought and happy to take any questions. Thank you.
I have time for one question before a break. Jamie.
What the nature of the yield increase was? Was it just the size of the seed, the weight of the seed, seed number, number of tillers? What was the nature of the?
All of the above.
All right. So good news is we're heading into a break, thanks to our break sponsor, Board Malts. If you want to continue to talk to Molly during the break, feel free to do that. Because we are a little off schedule now, let's go ahead and return at 10:45. Good news is I'm presenting next and I can catch us up. So the next we'll get back on schedule, I promise. And I'm excited to go talking about how we will hopefully be funding some of this work. Very much so, as he said, in a holistic way. So enjoy your break and come back at 10:45.
Good job.
Sorry, okay, longer.
Don't worry about it. Oh, perfect. Thank you.